Yes, good morning everyone, and thanks for the opportunity to give a talk here at this audience. I have to say that my background is in mathematics, so I hope you will forgive me if I say some things that are not that applicable for plant breeding, but I hope also some of the things that I will say are relevant to you as well. We'll talk about how we can bridge genomic prediction and crop growth modeling, also with high throughput data phenotyping, using a new technique, which is known from the field of systems and control, which to my knowledge, never has been applied in this field. So I would like to show the potential of this using some in silico data. And as you all know, the genotyping environment interaction is one of the larger challenges in plant breeding and modeling of quantitative genetics. Because different genotypes rank differently across different environments, which is something which is difficult to capture in the static genomic prediction models that we're using. And one of the ways to overcome this is to use crop models, but as was pointed out yesterday as well, it's quite hard to use these models, also because they rely on very specific hypotheses that are not always true, and not all dynamics are captured in those processes. So the question is there something else that can help us answer and help us to disentangle the GYE interactions? And I think one of the things that has most potential here is the field of high throughput phenotyping, because this basically gives us a lot of longitudinal data over the season, for example, collected by drones, by imaging, by sensors, which gives us time series of traits. They give some information about how the GYE is interacting with time. And as we also saw yesterday in one of the talks, I think by Steve Penfield, was that it's really different per time which genes, for example, have their expression. So it's really important to also involve the dynamics in your genome prediction models. So the question is, how can we leverage this high throughput phenotyping data in the models to achieve better models, actually, without having to rely on hypothesis-driven crop models. So if you want to combine genomic prediction and crop growth models, several approaches have been proposed. One of the classic ones is, of course, to use environmental covariates in your models, which was also presented in some of the talks yesterday. But what you often see then is that this is static. So you have to come up with some environmental covariates that are aggregated over time, in some sense, to capture the dynamics of the whole season in only one single value. And by that, you will, of course, discard some information because you have to summarize it in only one value. Then another approach that was recently proposed in some papers is to use genome prediction to predict the parameters that can subsequently be used in crop models. And then you just use genome prediction to predict the parameters, and then you use the crop model as a nonlinear mapping, basically, from the parameters and environmental time series to predict the crop trait of interest, so say yield, for example. But to do this, you need to calibrate the crop model, which often comes at a high cost. You have to collect a lot of data, which is very costly. So it's hard to do, actually. And then a bit more flexible approach is to use the crop growth models as some indication of your environment. So you can, for example, extract stress indices from your crop models. I think in one of the talks of yesterday, crop models were also used to extract some covariates that were used in the crop prediction models and the genomic prediction models. And for this, the calibration burden is a bit lower because you can also use generic parameter sets. But this still comes at the cost of having a static model, and you don't have really insight on what's happening in the traits. So that's, to sum up the limitations of existing approaches. We have genome prediction models. Those are often static, although there exist also temporal approaches, as we just saw in the previous talk. But those have limited interpretability, so we don't know what is actually driving the genetic differences. And these ignore the physiology. And then we have crop models, which often have a lot of parameters and are hard to calibrate, especially with the high throughput phenotyping data that's collected nowadays. And why is that? There's a few reasons. So first of all, there's a mismatch. So our drones fly over the field and collect, for example, at the NDVI. But that's a trait that is not classically modeled in the crop models, so we cannot actually use that to calibrate our models. So there's a certain mismatch, and the dynamics of those high throughput traits are often not that well understood. So we cannot really implement them. And also the crop growth models that are used are often discrete in time, which makes it very hard to do any analysis. So sensitivity analysis, uncertainty analysis, it's all very hard to do with the classical crop models. So to sort of overcome these constraints, what we propose is a new method, which comes from the field of systems and control, which is called SINDy, which stands for Sparse Identification of Nonlinear Dynamics. And the idea is rather simple. So instead of relying on predefined hypothesis-driven group models, we just rely on the data, and we use the data to extract the dynamics that are behind it. And we try to link that to the environmental time series that we see. And the result of this is a compact set of interpretable differential equations that we can actually use for making simulations. And at a later stage, what will be also an opportunity, which I think is more relevant for the community of plant breeding, is to use the coefficients fitted in these models as quantitative traits in genome prediction. But I will mostly focus on the first two steps now. So how does SINDy work? First of all, we have to do data collection, of course. So we have to collect both environmental time series and crop traits over time. So we let the drones fly over the field, collect traits like the leaf-area index, the NDVI, it can be anything. So we're not restricted by any physiological relations that we know. We just collect everything that we can measure, and we collect time series. That's the most important for different individuals over one growth season. And we also collect data on the environmental drivers. So we take the temperature, we take the soil moisture, et cetera, et cetera. And then we construct a library, and this library is the most important part, actually, because in this library we store all the knowledge that we have of the system. So if we know that certain photosynthetic relations exist, we can put them in the library. But you can also do this in a more data-driven manner. So we can also construct a polynomial library, include linear terms, quadratic terms, interaction terms. We can basically include everything that might be relevant. It doesn't have to be relevant, but just whatever we think could be relevant. And then the next step, that's actually where we select from the library of candidate functions which terms we want to keep. So we do sparse regression. We take the numerical time derivative of the crop traits, which is denoted by the x dot here. So the matrix X contains all the crop traits over time. We estimate a numerical derivative. And then we try to fit it to the library of functions, denoted by the theta times some matrix Xi containing all the coefficients. And the main thing about sparse regression is that we set this minimization problem such that the Xi matrix in this problem is sparse. So we only want a compact set of coefficients. Because then the output will be a compact set of genotype-specific equations that describe growth. So how does this look a bit more visual? So we collect our data on crop growth using remote sensing. We know that the growth is driven by environmental drivers. We also collect these time series. We store those time series and then construct our matrix and a vector equation. So here we have the matrix for the crop traits. We estimate a numerical time derivative using numerical methods. And then we, in this matrix, theta, we put all the relevant trait relations that we think might exist. And in the end, the SINDy approach selects the ones that actually are relevant for the current data set, which is shown by the highlighted columns here. And the matrix C contains the coefficients, which actually explain how much they contribute to the growth in the season. And this also can also be written as a system of differential equations shown at the bottom. So if we run this, what does it look like? So let's look at an in silico example. We look at a potato generated by Wofost. So we only use Wofost, which is a crop model, to generate the growth curves, and then we discard it. So we don't look at the equations in Wofost anymore. We just use it to generate realistic data. We generate data for 5 cultivars in 12 different years. And we look at the leaf-area index and the storage organ size, denoted by L and by S in the continuation of this presentation. And we also select the data for the environmental drivers. And then we select the libraries. So which libraries do we choose to model the functions? We select 3 different libraries. The first one is a driving-only library. So we only model the growth rate, the SDT, in terms of environmental driver functions, which can be nonlinear. So that's denoted by the V function. That's basically a basis function that can be nonlinear in the environmental covariates. But it's also important to note here that it's a linear combination of all the nonlinear functions in the environmental covariates. So the coefficients Xi are still put there as a linear combination. And those are the genotype-specific coefficients. So this equation is linear in the genotype-specific coefficients, but nonlinear in the basis functions in the covariates. Then we have another library that we use, the CMI-coupled one, and in this one, the difference is that we also put the L. So we use the L, which stands for leaf-area index, to give feedback to the growth of the storage organs, which of course makes sense from a biological point of view, but it adds some complexity to the library as well. And it's important to note here that the L is given as a measured input. So we don't model it yet, it's just a measured input. Then in the coupled approach, we model both the S and the L. So the nice thing about this approach is that we can model both the growth of the storage organs and of the leaf-area index, just as a function of numerical, of the time series of the environment, without any other inputs. Okay, so what does this look like in practice? If we run this for one of the time series that we got, then this is one of the equations that we get out of it for one of the genotypes. So this is an ordinary differential equation, and we can use this to model actually. So we can use this to generate growth curves over time that describe how the genotype responds to the environment in time series, basically. So we can also look at scenarios where we provide a part of the time series and see how this affects our genotype. And what we also see is that for both genotypes, we have a similar model. Many of the terms that appear in the one model also appear in the other, which means that the same environmental covariates and interactions play an important role for both genotypes, although the magnitude of the coefficients is different, which means there's genotypic differences, which we also want to see, of course. And well, let's look at the RMZ. So when we look at the error in the predictions, we arrive at predictions around 400 kilograms per hectare, which means that we have an error of about 4 to 5%, depending on the exact magnitude in the specific year. So let's look at some curves to see how this looks. So these are growth curves. The black dash curve is the one that's predicted by the crop model itself, and we try to recover. We see we can actually very well capture this curve. So we can capture the dynamics just by looking at environmental time series and the measurements of the storage organ size. So we can just basically recover the data using this system identification approach. And for leaf-area index, we can do more or less the same, but then it's a bit harder also because of the steep decay in the parameters that's not well captured in the environmental drivers. And if we want to predict yields, we see that we can achieve very high correlations, also because there's not a lot of error in this data set, of course. In reality, this would look different. Then one last result that I would like to point out is the magnitude of the coefficients across the genotypes. So every color is a different genotype here. And if you look at the first term here, the ILM, which stands for the degree days times the leaf-area index times the soil moisture, we see that all the different genotypes have a positive contribution of this term, but the magnitude of this contribution is different, which means that there's genotypic differences in the growth curves that we'll observe, which is clearly a clear indication for genotypic differences in response to this variable. So that's actually something that we would like to use in the future. So to give you some takeaways and an outlook, we propose SINDy as a new approach to turn high-throughput phenotyping data or other types of data where we have time series into a complex set of equations that can actually be used for modeling as well. Results on in silico data show good results, low errors of a few percent only. But of course, the question is how this translates to realistic data with sparse measurements and more noise. And also what we see is that the identified coefficient seems to be meaningful, so they are also consistent across genotypes. And then the outlook that we have is that we want to validate this, of course, on realistic data, and we also, at a later stage, want to integrate this with genomic prediction models, so see if we can treat those coefficients as quantitative traits in genomic prediction models. And the full details of this presentation will appear in a paper that will be published later, hopefully. And in the end of the presentation, I would like to acknowledge the PROPESAR program, which is the research institute that I'm involved in, which is a collaboration based in the Netherlands between many industrial partners and academia. And of course, thank my supervisors for their inputs. Thank you.